My name's Josh Morgan. I'm a postdoc researcher in Jeff Lichtman's lab at Harvard. We want to understand how the nervous system works by looking at how neurons organize their synapses with one another. So all behavior in animals depends on cells talking to each other by these specific connections. They send out long processes and form synapses with one another. Understanding the pattern of those connections is one way of understanding animal behavior. We use large-scale electron microscopy to reconstruct neural circuits. Large-scale EM lets you see every cell and every synapse in a circuit and find out how they're wired to one another. I've imaged a very large volume of mouse lateral geniculate nucleus with electron microscopy that allows me to see how hundreds of neurons are connected to one another. This data set was collected in the Lichtman lab where I work. One of the goals of this data set is to understand whether the neurons in the cerebral cortex selectively form synapses between particular pairs of cells or are kind of randomly wired. I just had a look at the data in this kind of three-dimensional viewing mode, and I was able to fly around and look at different spines and rotate around the spines. And I think aside from the fact that anytime you look at the data from a new perspective, you can see new things, I can imagine when you're editing cells and checking for mistakes in tracing, you'll be able to, follow a cell, look for anything odd, and see where things need to be fixed. So in the future, I could see this helping me to look at my data from a new perspective.